Hi, welcome to the second installment of my fourth lecture in my series on the coronavirus. In the first installment of this lecture, I did a compare and contrast exercise of the innate and the adaptive immune systems. In this installment, I'm going to delve more deeply into the innate immune system and how it works. Okay, some of this is ground we've already covered in prior lectures. So you have, over here, you have the endosome, and in some cases the virus enters the cell by first coming in through an endosome. And in the endosome, this virus has released its RNA genome, and this virus has burst open in the endosome, perhaps because there's enzymes in the endosome that were acting to weaken the virus. Now, cells have what I'll call molecular eyes. These are special devices that the cell uses to constantly scan the environment inside the cell for the potential presence of foreign invaders. And these molecular eyes look for features that would normally never be present in a typical human cell. And one of those features for the coronavirus is its RNA. So one difference between the viral RNA and normal RNA is that the viral enzymes that make the viral RNA are different than the ones that our human cells use. And in particular, the result of that is that the start of the viral RNA is chemically distinct. And so there's molecular eyes, for example, this one that's embedded in the membrane of the endosome, that can see that difference, and that switches on a signaling cascade. Meanwhile, if the viral RNA genome ends up in the cell, we already discussed that one of the first things that happens is you make this long polyprotein that gets chopped up, and the pieces that are released from that then form an assembly line to copy this viral genome. And so they take the positive strand copy and make a negative strand. And that leaves you with a very, very long, 30,000 bases long, double-stranded RNA. And again, that is something that's not typically found in one of our human cells. RNA can be double-stranded, but normally you don't have very long double-stranded segments. So you have molecular eyes in the cytoplasm, and they can see that very long double-stranded viral RNA, and that is a signal that something's happening here. All right, and it's probably not good. And so you have these signals that are initiated by these molecular eyes, and those signals can travel all the way to the nucleus. And when they get to the nucleus, they turn on the expression of genes in the nucleus. And I'll draw one example here where you have a set of genes that are turned on, and they produce RNA molecules that come back out into the cytoplasm. And those RNA molecules are then engaged by the cell's 3D printers, the ribosomes. And one of the proteins that gets produced, and I'll draw its shape like this, gets secreted from the cells. And this is a class of proteins known as interferons. And so there's multiple different interferons, and they do multiple different things, and I'll focus here on one particular class. So these interferons get secreted in response to the cell believing that it may be under attack from a virus. Now, what's the consequence of that? So imagine that this is a cluster of cells. This is a sheet of cells in your lung, okay, referred to as the lung epithelium. And imagine that one of the cells in this group becomes infected with a virus. So I'll crosshatch the cell to make that more obvious. And this cell then detects the virus using all the mechanisms I've just described to you, and it begins to produce interferon. And that interferon is secreted outside the cell. So I'll draw multiple molecules here coming outside. Now, a key point is that cells have interferon receptors. All cells have these kind of receptors. And I'll just draw a few of them here. And the interferons that are secreted can dock into these interferon receptors. And when they dock onto the receptors, that turns on signals inside these cells. And those signals do a few different things, and I'll just emphasize two things here. One is that it leads to the activation of an enzyme that destroys RNA. So these red squiggles here are RNAs inside the cell, so those are eliminated by that signal. Okay, so I'm just going to erase them with my finger here. And what that does, now remember, the RNAs are essentially the instruction set for the 3D printer. So they're the file for the 3D printer. So once you destroy those RNAs, the cell's not going to be able to make proteins anymore. Now, as if that weren't enough, there's a second signal triggered by this interferon, and that is that signal shuts off the ability of the ribosome to begin making new proteins. So I'll just draw an X through the ribosomes here. This is temporary, it's not permanent. It just shuts the ribosomes off for a while. So two things have happened. You've gotten rid of the file that the ribosomes use, the RNA that the ribosomes use to make proteins, and you've turned off the ribosome. So what that does is it shuts down the production of new proteins. Now that's okay for a cell in general because the cell already has pretty much the proteins that it needs. However, it's a real problem for a virus because for a virus to copy itself, it has to make new proteins, and it has to make a lot of new proteins. And so when you have this response, then you can't make new virus in the cells that have received this interferon signal. And so that buys time for the adaptive immune system to kick in and come and join the fight. Now, the interesting thing here is the interferon may be produced just by one virally infected cell, but since it's secreted, it can diffuse around its neighborhood. And so all the other cells in the neighborhood of that infected cell now shut down their protein production and become refractory to being inhibited by virus, or I'm sorry, become refractory to being infected by virus. Now, I'll come back in the next installment, in the third installment, and I'll describe another aspect of the innate immune response that plays a key role in helping to fight virus.